There is no arguing that genetically modifying human embryos is in the near future. The possibilities this would create for society are incredible. The science behind genetically modifying the embryos is pretty straightforward, as Caliph and Nalubala explained that the CRISPR technology trudges along the genome to edit the RNA sequence by omitting, rearranging, and adding genetic material as needed. In 2016, this technology was used to assist a couple in Mexico as they wished to have a healthy baby. According to Regalado, the procedure involved changing the genetic code to correct for a genetic abnormality which had the possibility of seriously harming the child or even causing the child's death. While the technology has shown to be useful and successfully implemented, there are still a lot of issues surrounding it. To begin, Ma et al. Says the efficiency or likelihood of the change actually occurring is due to not all cells accepting or receiving the change. It is possible for the human embryo to reject the genetic changes altogether. The best models of this technology have the efficiency around 52 to 72%. Even the models with higher success rates on animal genomes, where birth rates were only 10%, have efficiency issues with the human embryo due to the genetic structural differences, as found by Chen. Furthermore, before this technology can be made available to the public, more research needs to be done on the true effects of genome editing on the embryos as they are born and grow into adulthood. It is currently unknown if there would be adverse effects on development, and the chances of making an incorrect edit, thus causing exponential harm to the child or, once again, even death, are still high. The unintended harm can also be caused by the differences in the individual genome. While there is a base composite genome referenced, each person has subtle differences in their genomes, making them unique. Not accounting for this can lead to effects that are unwanted, unpredictable, harmful, and deadly. Editing the genome in the human embryo requires in vitro fertilization. This raises the cost of having the child and is likely to increase the risk of major developmental issues such as heart deformities and Down syndrome. According to the National Academy of Sciences, the FDA guidelines may be ignored by physicians in order to begin utilizing gene editing in clinical practice to treat genetic diseases and infertility. Barry Collar sat on a committee made up of members from the U.S. National Academy of Sciences and the National Academy of Medicine. In February of 2017, this committee decided on ethical standards for non-inheritable gene editing. Gene editing is only permitted in certain cases, and that would be to avoid the issues of ethics. The certain cases would be that it must be only to prevent the transmission of a serious or deadly disease or condition, or it can only be used on DNA strands known not to be associated with disease, and it must be done under strict ethical and legal guidelines. The ethics are incredibly complicated surrounding the issue. There are global guidelines, and each country has their own standards to be met. In 2015, the United States proposed a break on the research due to the ethical implications. The FDA even classified edited genomes as a drug due to the intention of disease treatment in order to put a halt on the research. Overall, the human genome is a tricky subject, both to physically manipulate and to work with legally and ethically speaking. While we are close to having this technology available, there are still many hoops to jump through and RNA sequences to revamp before the technology can be truly integrated into main societal use.